Hi everyone, my name's Alan Dorin. I'm a researcher at Monash University in the Faculty of IT, and I'm here to talk to you today about improving insect pollination in protected berry crop. You're probably wondering why a computer science researcher would be talking about such a subject, but I hope during this talk that you'll come to understand my interest in pollination. So I'm sure you're well aware, and I don't really need to tell you that pollination is a big deal, and it's a big deal not just for global food security, but more specifically within the berry industry. It's a big deal for many varieties to provide adequate crop yields and improve quality. How big a deal is it? Well, this diagram doesn't really do it justice. You could say it's this big, but still you'd probably be underestimating the amount of impact that pollination has on the berry crop industry and crop yields within that industry. Now, why? Why would I be focusing on this if it's known to be a big issue? Surely there's been plenty of research and we're at a good point with regard to our understanding of pollination of berry crops, and we know what we need to do to get adequate pollination for good yields. Might be a little bit surprising to at least some of you that that's actually not the case, and that there's a lot we can do to improve pollination of our berry crops. And here illustrated is one reason why. So I'll ask you for a minute to imagine you're a tiny insect about 1 or 2 centimeters long, and I blindfold you and I put you in the middle of a polytunnel that looks like this strawberry polytunnel you can see here. I spin you around and I ask you now to identify the location where you entered the polytunnel, and then to go and find your hive. As you can imagine, for a number of reasons, this is very difficult. Firstly, the insect can only see something like 75 centimeters, and these polytunnels can be 100 or more meters across. Secondly, insects such as the honeybee depend on polarization of the light for navigation, and the light is scattered and diffused by the polytunnel cladding. Another thing small insects such as honeybees use to move around and navigate successfully are landmarks. But no matter which direction I look in a polytunnel like this one, everything looks the same. There's the same repeated poles, the same repeated hoops, and so on. So by setting out our crops in environments like this, which are well suited, no doubt, to the plants, we're actually making things very difficult for the honeybees upon which we depend for pollination. So the projects I'm involved in try to understand why this is the case, like why are these environments so difficult for insects to navigate, and I've just given you a few examples now, but also what can we do to improve the situation, to improve their ability to move across the crops, to visit the flowers on the crops, and to pollinate them so that we produce good quality crop. So one of the projects I'll be talking about is a newly funded project with Costa Group and the Australian Blueberry Growers Association, as well as University of New England and Western Sydney University and CSIRO and Monash University, where I'm based. So the purpose of this precision pollination project is to change the way we think about pollination practices, and to do so in a way that is reusable and repeatable and based on sound site-specific data. So most of you are probably familiar with the kind of stocking rate approximations that you would get with various crops that might tell you you need this many hives for this many hectares of a particular crop. But what we're looking to do in this project is to generate data that is specific to a particular site. So that's particular varieties of crops, in particular landscapes, under particular polytunnels or other protected cropping and semi-protected cropping structures, laid out in particular ways at particular climates and so on. And as you can imagine, all of these factors impact on insect behavior, and therefore they impact on insect pollination. So using the kind of general cheat sheets that you get on pollination guidelines for hive stocking rates are a start, but we can do much better than that. And that's really what this project is all about. The project operates in a loop, a cycle of monitoring, forecasting, and management, which I'll describe in a little more detail throughout the rest of this talk. The aim is to generate a series of transferable mechanisms for monitoring insects, for modifying environments, and for making forecasts about what kinds of modifications are most likely to be successful before we need to intervene and try them. And the outcomes of the project, we hope, are improved crop yields and quality. And we've got experience from running this and a number of trials that make this a viable proposition. So how does the process work? As I explained earlier, it's a three-stage process that operates in a loop. So you can see here the three stages set out. The first one is the digital pollination monitoring. In this, we track individual insects and we collect data for a specific location within a polytunnel, and then we replicate that multiple times around the polytunnel. Once we've collected this data, we take it and sort of load it into computer simulations to adjust the simulation behavior to specifically match the particular site that we're studying. So we're not building generic simulations here, we're building a simulation of insect movement on a particular crop at a particular location. After we've built these simulations, one of the outputs, the results from this simulation, are a set of recommendations or suggestions that provide advice or ideas on how we might reconfigure the environment in order to improve pollination in various ways. And we work with the growers and the beekeepers to decide which of these are viable, and then we intervene in the crop, adjusting some parameters perhaps, and then we go around the loop again and we re-monitor. And hopefully after we've been around this cycle once or twice, we're seeing higher yields and better quality of fruit. Now I'm going to talk about each one of these three steps in a little more detail to give you a more complete understanding of what we do. So the first step, digital pollination monitoring, involves us stationing a very small series of computers. They're about the size of a couple of mobile phones strapped together, I suppose, or a cigarette pack. We position these devices around the polytunnels at various locations. Now, each one of these is capable of recording digital video of an area about 60 centimetres by 40 centimetres at a resolution that's capable of enabling us to pick out individual insects visiting individual flowers. We can leave these devices in situ for hours each day, for days each week, and for several weeks of each growing season, depending on the needs of the site and the managers of the property. Of course, we've got to be careful that they don't get in the way with the usual kind of farm activities going on. So depending on how we set things up, we may need to remove devices at the end or beginning of each day or so on, depending on the particular requirements of the location. So you can see in the images here, again, this is the strawberry crop, just a sample of some of these devices set up in a grid in this case. And you can see at the bottom right, one of the images with a scale position just for clarity, showing what each one of these cameras sees and then records. So once these are installed, they capture continuous data. Now, if you have a look on the left, this isn't the strawberry crop anymore. This is some wildflowers. You can see just at the right-hand side, a little bit of movement and a few red dots appearing. So this is the location that some artificial intelligence software has identified the presence of a bee. In this case, it's a honeybee. And the software automatically identifies those insects even amongst the complex foliage, even when the foliage moves in the wind. And it picks out the insect's path and it records that and documents all the flowers that this insect has visited. It documents the amount of time each insect spends on a flower. It documents the order in which it visits flowers, and from that we can understand the pollen flow. And it generates a series of these trajectories or paths across the image, regardless of whether or not the insect is walking or flying or remains stationary. Now, from a series of these videos, we build up a heat map, like you can see in the top right-hand corner of this slide. So this heat map shows us the regions of the crop that are visible to the camera that are most frequently visited by insects. And we can estimate from this, for instance, where there might be limited numbers of insects visited, but also where there might be areas that are very well populated. Now, you might have noticed in the video on the left there, a second bee has just entered and it's not currently being tracked by the software. And now there's a third bee. It's not currently being tracked. That's simply because this video is showing an early version of the software that was only able to track a single insect at a time. The latest version of this software is able to track any insect simultaneously with all others in the shot. And what we do from this data then is build up a series of paths, as you can see at the bottom right here. Each one of these paths corresponds to a honeybee track in this instance. The blue circles correspond to flowers, depending on the crop. And we go about then building up an understanding of how the insects are moving and engaging with the flowers across the entire area that we're monitoring. Here's a whole polytunnel illustrated at the top right. So in this particular polytunnel, which was strawberries, we stationed 9 monitoring stations throughout the crop. And you can see the data from each one of those indicated in the blue squiggly lines in the middle. So the blue lines represent honeybee tracks. And you can see in this case, we were also interested in other insects. So we've trained our artificial intelligence software to recognize not just honeybees, but in this case, hoverflies, various types of moths, butterflies, and even some wasps. And we're able then to work out not just which insects are present in the crop across full days or even weeks, but which insects are most frequently visiting the flowers, how long they're spending on the flowers, how many of the flowers have been successfully pollinated, which is based on our understanding of what type of visits and what length of visits are required to pollinate a particular crop flower. So we get a very complete picture then of what's going on out in the field. Now, as any beekeeper will tell you, of course, it's important for us to be able to relate this to what's going on at the hive. So in addition to monitoring what goes on in the field, we also are developing new technology to monitor the insects' movement and behaviours in and around the hive. Now, this example here is actually for native Australian stingless bees, not honeybees. But you can see here the algorithm in the middle is identifying insects, tagging them, and then following their movements, from which, just like on the field, we can extract information. This is work in progress. Please don't tweet this one. It's being carried out at my Native Bee Plus Tech facility at Monash University in the Faculty of IT. So once we've collected this data, that completes stage 1 of the cycle that I was talking about before. Stage 2, as you recall, involves generating computer simulations. Now, these computer simulations you can think of a little bit like a computer game. Say, for example, Pac-Man, with which I'm sure many of you are familiar with. If you imagine the ghosts moving around on the maze in a Pac-Man game, but now switch the ghosts for honeybees, let's say, and switch the maze for cropping environments, then you've got the right idea. So these individual insects are monitored in simulation as they move around a virtual environment. And their decision-making and their movements are based on what we know about insect, in particular bee color perception and preferences, their visual range and acuity, their sense of smell, their memory, and their decision-making. So we build up these kind of sophisticated models of how insects think and behave, and we use those models then to drive virtual bees around virtual foraging environments. Now, Each one of these bees, if you like, has a brain that can be represented as a flow chart like these two here, the one on the left for a honeybee and the one on the right for a bumblebee. But each species of insect has its own unique behaviors and physiology and also learning capabilities and many other things. So we need to be quite specific in the way we generate these flow charts that represent and dictate the way our virtual bees will think. Once we've built those, what we do is we build a virtual world that mimics the environment that we want to simulate. So you can see 3 examples here. On the left, we were building a simulation that explores the impact of subdividing small plots of land into single crop varieties, such as you might find in some subsistence farms or in community gardens in developed nations, major cities. In the middle, you can see a more traditional industrial crop layout. On the right, we were looking at crops interspersed with weeds or meadows in which there were, say, multiple species of flowering plant intermingled with one another. So these virtual environments then become the home for the virtual bees, and we monitor the pollination that we get as we adjust the conditions and the insects' behaviours in the landscape. So here are 3 examples that we've just documented for the virtual environments. On the left, you can see the typical kinds of plots you might find in a community garden. This one's from Edinburgh in Scotland. In the middle, you can see some potato fields from the Netherlands. At the right, you can see a rural landscape in a developing nation planted in very small plots. So what we might want to know in all of these scenarios is, on these particular sites, how do the insects behave? So it's not just a matter of saying, well, I've got this crop here and I've got it in these 3 different environments, and then expecting that the insects are going to behave the same way in these 3 environments just because it's the same crop. That doesn't happen. We know that doesn't happen. So what we want to do is understand how the local site-specific conditions can impact the behavior of the insects. And that's one thing we can try out in simulation. Another thing you might be thinking about is, well, how can I adjust the movement of insects in my industrial site, such as to improve, say, cross-pollination? So you might be wondering, well, how can I plant different varieties of my, say, strawberries, as you can see here, to improve pollination, pollen flow between variety B and variety A? And you might be aware, for instance, that insects typically travel along rows of crops like this. But obviously, that's not going to help you if you want pollen to flow from variety B to A, if the insects continually move along variety A's row. So our simulations can enable us to suggest alternative planting structures or ways to change the environment or position the beehives, perhaps, that will improve pollen flow in the way that you desire. Here's a simple case study we conducted also with strawberries and raspberries. In this case, what we found is way more honeybees in the raspberry than the strawberry. So as soon as these 2 species of crop were co-flowering, the bees went straight to the raspberries and ignored the strawberries. One thing that we also discovered, if you look at the green square labeled 10 in the middle there, is that the number of bees on the weeds at the bottom left of the figure on the left was something like 10 times as much as any of the bees found on the strawberry crop. So a simple suggestion we could make in this scenario to the growers, well, if the strawberries are flowering, now's probably a good time to mow your weeds, because in fact, the hives that you've located within the weeds are primarily foraging from the weeds, and they're not visiting the strawberry crop as you would like. As I mentioned before, we can use the simulations also to help us with site-specific management, moving hives, adding hives, removing hives, and adjusting the planting arrangements of the crop. Here we see a hybrid seed production facility, and similarly, we're looking at how to align the crops to maximize pollination of the female lines whilst minimizing the number of male lines that are planted. So that just about sums up the way this process works. As I said, it's a repeated cycle. We don't just go through it once. We work very closely with the growers and the beekeepers to make sure that the recommendations that we suggest are useful and that they feel that they're actually manageable before we proceed with anything. Now, I'm quite open to questions, but obviously I'm not with you. However, you can see my email address there and my Twitter handle. If you have any pollination issues you'd like to discuss, then please get in touch with me. I'd love to hear from you. Thanks very much for your attention.